Now that we did a little review, we're going to talk about the exemption of the Mendelian principles, and sometimes we will give you some examples and that uh, the traits are not dominant or recessive, and it looks more like a blended inheritance that we talked before. However, as I will repeat this multiple times, these examples are not saying that genes blend, that this mechanism is right. What they are saying is that Mendelian inheritance work perfectly, and if you understand Mendelian inheritance, you will be able to understand why you get a pink here. Okay. Also, another proof that Mendelian inheritance is that you have these multiple alleles that also show the mechanism of the Mendelian inheritance is right. However, not always we are going to find this dominant or recessive. We can find multiple alleles, and we will talk about these ABO blood systems. And finally, one example that is more complicated is this, that is also very common, is these polygenic traits. So polygenic, poly means many. So you have many traits that are involved in only one phenotypic trait. And one example here you see is the height, and the height is determined by multiple genes involved. In addition, as I said to you, the environment also plays a key role in many other traits. So you can have polygenic traits too, and you have also the influence of the environment. So although we are completely sure that Mendel is right, Mendel is correct, like Darwin is correct, however, they cannot explain all the phenomena. Yeah, and we're going to show more complicated examples that are exception of the Mendelian rules. Now, so we're going to start with the first one, that is the incomplete dominance. And to do that, I want that you start watching a video, again, of the Campbell biology that show the example of the aliens that we have been talking in the previous chapters.
Incomplete dominance. Let's examine how the horn trait is inherited in Mendelians, a mythical alien species. If we cross an alien with 2 horns and an alien with no horns, what do you think the offspring will look like? Let's find out. The F1 offspring all have 1 horn. Note that the phenotype of the F1 generation falls in between the phenotypes of the 2 parents. If we cross 2 F1 individuals, what do you think the offspring will look like? Let's make the F1 cross. For the F2 generation, we observe 24 Mendelians with 2 horns, 47 Mendelians with 1 horn, and 22 Mendelians with no horn. The F2 generation exhibits a phenotypic ratio of 1 to 2 to 1. This is different from the ratio of 3 to 1 in Mendel's pea plants. In Mendel's peas, the dominant allele had the same effect whether an individual had 1 copy of the dominant allele or 2 copies. What do you think is going on with the horn trait in Mendelians? The simplest hypothesis is that the horn trait exhibits incomplete dominance, which means that neither the 2-horn allele nor the no-horn allele is dominant. To understand this trait, let's assign genotypes to the parental cross. Since neither allele is dominant, we can't use capital and lowercase letters to indicate the alleles. Instead, we'll use H for the horn trait with a superscript T for the 2-horn allele. Thus, the homozygous Mendelian with 2 horns has the alleles H superscript T and H superscript T, and produces gametes with H superscript T. We'll use H with a superscript N for the no-horn allele. The homozygous Mendelian with no horn has the alleles H superscript N and H superscript N, and produces gametes with H superscript N. The F1 generation inherits the alleles H superscript T and H superscript N. This heterozygous genotype gives the intermediate phenotype of 1 horn. In general, if 2 homozygotes are crossed that differ in a trait, and the offspring all have a phenotype intermediate between those of the 2 parents, then the trait exhibits incomplete dominance. Now let's assign genotypes to the F1 cross. The F1 Mendelians with one horn produce the gametes H superscript T and H superscript N. We see the results of the F1 cross in the Punnett square for the F2 generation. The H superscript T genotype produces the two-horn phenotype. The H superscript T H superscript N genotype produces the one-horn phenotype. And the H superscript N H superscript N genotype produces the no-horn phenotype. In incomplete dominance, heterozygotes have their own unique phenotype, so the phenotypic ratio and genotypic ratio for the F2 generation are the same, 1 to 2 to 1. Now let's investigate the genetics of head shape in Mendelians. We begin with a cross between two Mendelians with round heads. The resulting offspring consists of 8 Mendelians with tall heads, 15 Mendelians with round heads, and 7 Mendelians with wide heads. What's the simplest explanation for the inheritance of these head shapes? The offspring are in a 1 to 2 to 1 phenotypic ratio, which is the characteristic ratio expected for a cross between two heterozygotes if incomplete dominance is involved. Thus, the simplest explanation is that these head shapes involve incompletely dominant alleles, with one type of homozygote having the tall head shape, the other homozygote having the wide head shape, and the heterozygote having the round head shape. If we look at the genotypes of the parents and offspring, that will help us understand the inheritance of this trait. Let's use S for the head shape trait, with superscript T for tall head and superscript W for wide head. The round-headed parents are heterozygotes, S superscript T, S superscript W, so half the gametes are S superscript T and half are S superscript W. In the Punnett square of the offspring, we see a ratio of one tall head, S superscript T, S superscript T, to two round heads, S superscript T, S superscript W, to one wide head, S superscript W, S superscript W. What offspring would you expect from a cross between a Mendelian with a round head and a Mendelian with a wide head? A Mendelian with a round head has the genotype S superscript T, S superscript W, so the gametes are S superscript T and S superscript W. A Mendelian with a wide head has the genotype S superscript W, S superscript W, so it has only one type of gamete, S superscript W. The offspring are therefore in a ratio of one round head, S superscript T, S superscript W, to one wide head, S superscript W, S superscript W.
Now we're going to talk about the different type of dominance. And as you watched in the previous video, we're going to talk about the incomplete dominance. Okay, now we're going to talk about incomplete dominance. And to do that, I'm going to show you this schematic that you can see here that explain both concepts, incomplete dominance and codominance. While in the incomplete dominance, the traits blend. So for example, if you have flowers that are pink and red, and flowers that are white, the offspring will be flowers that are pink. So here the two types of flower and the results will be pink flowers. So here is the definition. And incomplete dominance is when the phenotypes of two parents are blended together to create a new phenotype. And for example, when you have white flowers and a red flower producing a pink flower. And codominance is relatively similar, but here instead of having to blend the traits, here we have two discrete traits that are expressed at the same time, and in this case they're going to produce also a pink one, but in this case the codominance is when the two parents phenotypes are expressed together. So they are not blending, they are just expressed at the same time. And in the following example, I'm going to show you, in the following slide, I'm going to show you some examples. However, I highly recommend to go to the Campbell biology. They explained it very well there. And they show you that actually when we start defining these terms of codominance and incomplete dominance, we didn't know well the genes. Now that we go and know the mechanisms, we we are still we doubt a little bit. We have questions about what is incomplete dominance and what is codominance. When we go and see what are the genes involved, what are the proteins, what are the real mechanisms. So there is a line that is not as clear, but I'm going to, in the test, I'm going to show you some examples that they differentiate them, but please go and study the camel biology. Now I'm going to give you more examples of the incomplete dominance. So first thing that I want that you see, that you remember, is that the incomplete dominance, in this case, you will have that every genotype has its own phenotype. And you can observe that here. You can have that the homozygous dominant has its own phenotype. Here you can see that the homozygous recessive here and here has its own phenotype, and you can see here that the heterozygous has its own phenotype. So these three. Now, another important trait of the incomplete dominance is that either of the alleles is not complete dominant over the other one. So here you have the red allele or the white allele for the color of the flowers, and both none of those is complete dominant. So you will have these pink flowers, and finally, when you have these blended phenotypes, you will create a new you will have a third phenotype. So in other words, you will have two alleles, but you will have three phenotypes. And this is different from the previous examples of Mendelian inheritance that you generally have. When you have two alleles, you will only have two potential phenotypes. So heterozygous phenotypes are somewhere in between the two homozygous. So here I explained that red, white, that they were the homozygous, and in between, as you can see here, in between the homozygous and the other homozygous, you will find the heterozygous that are pink in this case. And of course, I'm showing you this with this simple example of the flowers. There are multiple examples, and you can see them in the Campbell biology. So let's now solve a question. And here they ask you the following question. They ask you to show the cross between a red and a white flower. And this will be very easy. If you want, you can skip this section. However, if you have any question, I'm going to explain this again very slowly. So first thing that you need to know to have clear is that which trait you're going to study. And in this case, you're going to study the flower color in this cross, which phenotype. So you're going to have a red and a white flower. And as a result, since incomplete dominant, you're going to have a third phenotype that is the pink color. The third thing that you need to know to solve this question is to know the genotypes. So the homozygous will be two capital R's. The pink, it will be the heterozygous, and the white one will be the homozygous recessive. In case that I made a mistake in the slides, I have been checking. Sometimes PowerPoint autocorrect and give here, make it a capital. I try to correct it as much as possible. If one day if in one case I forget, please send me an email and I will remind you. But it's not because it's just because PowerPoint keep constantly autocorrecting some of the slides. So remember, every time that we have a homozygous recessive, we should have two lowercase. Okay, now let's go. Now that we have all these three things clear, the trait, the phenotype, and the genotype, now we can draw the cross. So we will have a red one, and we will cross it to a white one like this. So that will be, and here I'm showing the cross between the red one and the white one with both with phenotypes and genotypes. Now that we know that, we can move and again get the ratios and the percentage of the genotypes and phenotypes. Again, this is very simple. So maybe if you study biology, you can skip this part of the video. But if you have any doubt, I will go again very slowly because I know that some of you don't remember your genetics. So just in case that you're one of them, you can watch the entire video. So again, the red one is here. Why? Because it's a homozygous dominant. However, the white one, it will be here because it's a homozygous recessive. So the two of them are lower cases. And here you will get, as a result, this heterozygous. This one will be also heterozygous, and this one will be also heterozygous. So in this case, 100% of them will be heterozygous, and all of them will have this heterozygous phenotype. Now let's go and see the phenotypes. So in this case, all of them will be, the percentage will be again all of them pink, and the ratio in this ratio, all of them will be pink too. So that was pretty simple. Now I want that you also try to solve this exercise that is very easy. Try to answer this question, that is, show the cross between pink and a pink flower. So what happened? What are the genotypes and the phenotypes, the ratios and the percentage when you cross these two heterozygous when it's incomplete dominance? And again, I will explain this in class, or I will post a video about it. So if you have a doubt, you can check it.